I will be speaking, yes, about caribou in west central Alberta and eastern BC, and I was already introduced. I have an ongoing project, and this is on evaluating genomic diversity for caribou in Alberta to allow for biodiversity monitoring, augmentation, and conservation. However, this genomic project just started. I wanted to mention it here because genomics, as opposed to genetics, will revolutionize much of what I'm talking about today. Specifically, I want to give you the heads up. So far, we have focused on patterns that may not be adaptive for the species. And in the future, we will know not only, for example, which are the population of caribou, but which population holds some traits that if those populations go extinct, we will lose for good. The project is funded by PTAC, NSERC, and other minor funding, and you will find all of these in the final slides with their logo. Today, I want to provide an update of some results that are relevant here. And I also want to mention that, yes, I got an email from PTAC, from Laurie, indicating that I should put some effort into figuring out what might be best management practices and also some costs associated. And I spent a bit of time on that. And this is a matter that will be presented in two slides towards the end. So today I will be speaking about environmental and anthropogenic drivers of connectivity in caribou. This is a paper that is in press in an interesting journal called Evolutionary Applications. Basically, it deals with implications of academic studies like this one. And please notice that the word caribou doesn't even appear on the title. This means that our results are applicable to other threatened species too. And we have figured out some information helpful to prioritize conservation efforts for threatened populations in general. So let me jump to the results, the study area, everything. So here is a study area. I told you West Central Alberta and Eastern BC. These are the contours of the populations that we deal with. And as you can see, they range from Banff National Park to Narroway. I don't know if you know, but yes, I'm sure you do, that there is no caribou left in Banff National Park anymore. The population got extinct. However, what this map represents is not only the populations, not only the samples that we got of caribou, which are these dots, but also the connectivity patterns and the direction of the gene flow. As you can see, there is some indication that we're dealing with a meta-population. And this is the actual data. It is basically indication of gene flow directionality from the vertical column to the horizontal column. And you can see that there are some significant signals of connectivity. For example, linking the Tonkin population of Jasper to the Brazeau, and then to the Banff National Park population. So this was a pattern of connectivity that was there in the past. And therefore, the gene flow in the past was directional from Tonkin to Brazeau to Banff National Park. And other connectivity patterns are here indicated in green, and you can see that the Tonkin, the Little Smoky, and the Red Rock Prairie Creek population that were all contributed to the AUPRF. And the Red Rock Prairie Creek population was also contributing to the Narroway. And basically, on the landscape, this made sense. So I told the Tonkin to Brazeau to Banff National Park. It's kind of interesting because these genetic results reflect the reproduction, so the past generation. So in the past, yes, caribou could move to Tonkin, from Tonkin to Brazeau to Banff, but that got extinct. And also, there are some GPS collars on this population, and really, dispersal between these populations is not detected recently. So we might be losing some connectivity patterns that were there until recent. On the landscape, again, you can see that there was some connectivity between Little Smoking and Alberta. I think it's still there. However, it is important to notice that unless we will resort in the future to drastic management measures such as relocation, we should maintain these patterns of connectivity. So what are these barriers to connectivity? Over here you see a map of the same study area, and we also mapped human features, such as, for example, roads, linear features, and forestry cut blocks. And here is the contours of the populations that we're dealing with and our samples too. We also work on habitat suitability, and we obtain a certain selection function for habitat of caribou. We use the inverse to see what is the friction of the environment, what is the difficulty in caribou from moving from one location to the other. And finally, we also looked at the predation risk. Now, the work of Nick, I say De Cesare, but I would suspect you would say Di Caesar, right? However, the name is of Italian origin too. And yes, Nick did a resource selection function analysis across scale for caribou, which is represented here. And this is the inverse. And of course, it doesn't include here human features or so. This one is everything except for wolf predation risk. This one and human features and so on. So they're mutually exclusive. So we could compare them, right? And what you see here is the least cost path. In other words, the effort that a single caribou would undertake to move from one point to the other to incur less predation risk, and you can do the same for less unsuitable habitat or less human features. All this allowed us to compare models of connectivity. So what were the barriers to connectivity? And we also did a comparative analysis, okay? So we had a number of factors potentially impeding connectivity, and we looked at which one was more stronger than which other ones. And these are confusion matrices. So the columns indicate principal models, and over here you have models that had vegetation, snow cover, elevation, other factors, right? And then we saw which model was beating the other model in so-called causal modeling approach. And you have here two columns that win when they are run against the other ones. And the two columns that win are distance. Of course, two populations will be more isolated if they're distant, but also unsuitable habitat between them. And then there was a third one that was also strong, which was predation risk. But as you can see, the red is stronger and beats all the other one, and this consistently ran along the first two columns. And again, it is distance and unsuitable habitat between populations. So what was this representing? This represented the likely cause of genetic isolation of populations. Now, again, to recap, two models were strongly supported. One was distance, and the other one was habitat and suitability. And again, I'm recapping a bit because I want to bring you to a certain point, okay? So what can we do with distance? Can we bring population closer? No, we cannot. But for habitat, maybe we can do something. And a model which was also supported was predation risk. So the more the predation risk between two populations, the more these are isolated. So in sum, before getting to the best management practices, our lab gathers and analyzes empirical data relating caribou to human impacts, describe patterns of connectivity to be maintained, and also manageable factors of isolation. This is why we work with PTAC, and we need PTAC. In general, we translate academic knowledge into applied products, and therefore, let's focus on the next points and next slides. And other work that we conducted clearly indicate genetic isolation of populations and also an interesting and strong correlation with population size. So there is genetic isolation, and populations that have less number of caribou, they are more isolated. How do we promote connectivity? Again, we cannot do much to address distance between populations, the first point. However, we could manage habitat between ranges. So perhaps we should think for the future of not just improving habitat within caribou ranges, which is this my understanding. Now, you are the expert, but it is my understanding that this is the current accepted approach. We should also look at habitat between populations. And this is an example of habitat suitability assessment. It is the already cited work by Nick, D. Caesar et al. I'm the last author because I was also the co-PI on the project. And it is published on ecological applications. So again, the main paper that I'm describing is evolutionary applications. This paper is ecological applications, and it highlights what are the factors of habitat suitability that we should manage for, right? Now, Laurie, in her email, she also mentioned to start thinking about the so-called environmental costs. This is how she defined them in the email. So I gave a bit of thought to that. Of course, you know, there are topographic factors that are important for habitat suitability for caribou. We cannot do much to correct those, like elevation slope aspect and where water is, right? However, we could invest some money, perhaps comparatively little money to the other ones, to assess these between populations too. There are some climatic factors, such as, for example, snow cover or distance to tree line. And again, there might be a correlation to climate change. Again, assessing might be relatively cheaper. Managing climate change might be very, very costly. This is why the $4 signs over here. And then there are vegetative factors, specifically land cover types, focusing on land cover types that are selected by caribou, also between the population ranges. And again, since these are areas of intense forestry, there might be a significant cost associated if we want to correct that. And then there are anthropogenic factors, and I don't underestimate the cost of rethinking those, okay? So there are $3 signs associated, and specifically, factor of unsuitability are cut blocks and also linear features. And on that, I want to conclude. I think I'm on time, you see? And with some photos of caribou, all taken by an incredible photographer and collaborator, Mark Bradley. And this is the real caribou that we are dealing with, and the organization that I need to thank.